Good afternoon, or morning, or evening, depending on when you are watching this video. So today we're going to continue our tour around the cell. So we're going to pick up where we left off and talk about the mitochondria, the chloroplast, and the peroxisomes. So the mitochondria is the site of cellular respiration, which is a metabolic process that generates ATP. So this is where you break down carbohydrates such as glucose. When you do, it lets off energy that can then be used to make ATP, which is the energy currency the cells use to perform work. So chloroplast is found in plants and algae and is the site where photosynthesis occurs. And photosynthesis is kind of the opposite of what happens in the mitochondria, and this is where this chloroplast takes solar energy and uses it to convert carbon dioxide into glucose or other saccharides. And there's also the peroxisomes, which is where oxidative organelles occurs, and this is where a lot of dangerous chemicals or chemical reactions occur inside the peroxisome, so that way it doesn't damage the rest of the cell. So the mitochondria and the chloroplast are not part of the endomembrane system. They have a double membrane and have proteins made up of free ribosomes. They also contain their own DNA. So if we look more specifically at the mitochondria, so the mitochondria are in nearly every eukaryotic cell. As I said, they have two membranes. So the outer one is a smooth outer membrane, while the inner one is highly folded into something called a cristia, which you can kind of see here, or you can see in the next slide. So the inner membrane creates two compartments. There's the intermembrane space between the outer membrane and the inner membrane, and then there's the inside both membranes, which is called the mitochondria matrix. And some metabolic steps of cellular respiration are catalyzed in the mitochondria matrix, and some occur in the inner membrane space. So the cristia, or the folding of the inner membrane, allows for a much larger surface area, and this allows for more enzymes that can synthesize ATP to exist. So if we look at an image of a mitochondria, you can see over here on the left-hand side an artist's description or depiction of one. So you have the outer membrane, which is fairly smooth, and then you have this inner membrane, which is highly folded. You can see there's a number of ribosomes as well, and there'll also be a plasmid of DNA, or a circle of DNA inside the mitochondrial matrix. Over on the right hand side, you can see what a TEM image of what a mitochondria actually looks like. And so you can see again the matrix, which is the inner inside both of the membranes, and then you have this thinner inner membrane space between the two membranes. So chloroplasts, which capture light energy, is a member of a family of organelles called the plastids. And a chloroplast contains the green pigment chlorophyll, as well as enzymes and other molecules that function in photosynthesis. So other plastids don't contain chlorophyll, and so usually are not green. Chloroplasts are found primarily in the leaves and other green organs of plants and in algae. So when you see a plant, if its stem is green, it likely has chloroplast in the stem. So similar to the mitochondria, there are a dual layer, however, of membranes, but the membranes in this case are slightly more similar to each other. You have both outer membranes being fairly smooth and the inner one being smooth as well. And then you have thylakoids, which is a membranous sac stacked to form a group of granum. And then you have the stroma, which is the internal fluid. So the stroma is similar to the chloroplast. So the chloroplast is the liquid which is inside of a cell and outside of any organelle. The stroma is inside the membranes but outside of the thylakoids and granum. So if we look at a depiction, here on the left hand side we have the artist's rendition, and you can see the inner and outer membranes are fairly similar. They're both kind of smooth and circle the stroma and thylakoid membranes. So then this fluid-filled area, or this empty space, would be the stroma, which is be fluid-filled of mostly water. And then you have these stacks of granum, which are part of the thylakoid membrane. And this is where photosynthesis occurs. This is where the enzymes and proteins reside that harvest light energy. And then over here on the right-hand side, you can see a TEM image of a chloroplast. And again, you can see the stacks of granum all connected as part of the thylakoid membrane. So peroxisomes is where oxidation occurs. So peroxisomes are specialized metabolic compartments bound by a single membrane. So unlike the chloroplast and the mitochondria, there's only one membrane. Peroxisomes produce hydrogen peroxide and convert it to water. So again, hydrogen peroxide is fairly dangerous to the cell, and so it occurs within the peroxisome where it can be taken from a dangerous state into water, which is non-dangerous, and so it doesn't react with any other parts of the cell. And oxygen is used to break down different types of molecules. So again, oxygen, which is where oxidation comes from. Oxidation means that you're adding oxygens. So this is where these reactions occur, which can also be dangerous to the cell. So if we look at a image, you can see here a peroxisome in the center with this large crystal of enzymes that break down peroxisomes, or break down hydrogen peroxide. You can see next to it, you can see a mitochondrion, so it's significantly smaller than a peroxisome. And then you can also see two chloroplasts, which are also significantly larger than both the peroxisome and a mitochondria. So this kind of gives you a set of scale of how large these three organelles are to one another. So the last component of the cell that we're going to talk about today is the cytoskeleton. And this is a network of fibers that extend throughout the cytoplasm. And it organizes the cell's structures and activities, anchoring many of the organelles. So it's a composite structure of three different types of molecules: microtubules, microfilaments, and intermediate filaments. And so this is basically the railroad system, or the walkways that connect all the different organelles within the cell. And not only are they the pathways, but they are also the anchors that kind of hold them in place, or the tether that can move the organelles around in the cell. So if we look at them in kind of more detail, you can see in this empty space, which would be the chloroplast, there's actually a fibrous network of filaments, of microtubules and microfilaments that attach to different organelles and the plasma membrane, and this kind of moves and shuffles the organelles around. Alright, so just to give you a brief overview of what we've talked about so far, we talked about the nucleus, which is where DNA is kept, and it has the double-bounded nuclear envelope on the outside. Next to that is the ER. Within the nucleus is where ribosomes are synthesized. So if we look outside of the nucleus, we have the endoplasmic reticulum, both the rough ER, which is studded with ribosomes and is this primarily site for protein synthesis, as well as the smooth ER, which does not have ribosomes and is primarily a synthesis of lipids and metabolisms of carbohydrates, and as well calcium is stored. You also have the Golgi apparatus, which is the sorting facility and package facility of the cell. So it takes in components from the endoplasmic reticulum and then modifies them and packages them and then sends them off towards the final destination. You have the lysosome, which is the digestive tract or garbage chute of the cell, and this is a vesicle which is filled with enzymes that can break down other proteins or DNA or saccharides. You also have the vacuole, which is this large open space here. You can also have, plants have the central vacuole, which is a larger vacuole which is filled with water, and is why plant cells are hundreds of times larger than animal cells. And then you also have the mitochondria, So this is the powerhouse of the cell, and while cellular respiration occurs, you have the chloroplast, which is the site of photosynthesis, and both the mitochondria and the chloroplast have a dual membranes. And then you also have the peroxisome, which is where oxidation occurs, and this is where dangerous chemicals are broken down, and the byproduct is oxygen. Sorry, the byproduct is water from hydrogen peroxide. So again, here's the nucleus connected to the ER, and this is where ribosomes are synthesized. And then the endomembrane system, which includes the endoplasmic reticulum, the Golgi apparatus, lysosomes, vacuoles, and other vesicles. And then you have the mitochondria, the chloroplast, and the peroxisomes, which is where cellular respiration, photosynthesis, and breakdown of hydrogen peroxide occurs. So now we're going to focus specifically on mitochondria and focus on cellular respiration. So living cells require energy from outside sources. Some animals, such as the giant panda, obtain energy by eating plants, and some animals feed on other organisms that eat plants. So these would be consumers because they cannot produce their own energy. So energy flows in an ecosystem as sunlight and leaves as heat. So we've talked about this in the previous chapters, but photosynthesis generates oxygen and organic molecules such as sugars, which are then used in cellular respiration. So cells use chemical energy stored in organic molecules such as glucose, which are generated through photosynthesis, to regenerate ATP, which powers work. So animal cells use glucose to generate ATP, and ATP is used as the currency to perform work within the cell. Plants can take oxygen and sunlight to make organic molecules such as sugar, which are then broken down in cellular respiration. So if we look at an image, you can see that light energy comes in to the chloroplast. Photosynthesis occurs within the chloroplast, taking carbon dioxide and water, and generates organic molecules and oxygen. And again, these organic molecules can be glucose, over here on the right. These organic molecules are then taken into the mitochondria, and actually so is oxygen also taken into the mitochondria, for cellular respiration in the mitochondria. So this is where the organic molecules are broken back down to CO2 and water to generate ATP. So you can see that this is a cycle between photosynthesis and the mitochondria that we rely on one another. The products of one, so products of photosynthesis, which are organic molecules and oxygen, are the reactants for cellular respiration, so we use these as the reactants. The products of the cellular respiration generates carbon dioxide and water, which is then used as the products for photosynthesis. And you can see that this is an endless cycle of generating products and then reactants to generate ATP. And then ATP is what cells use to perform work, and eventually the energy is lost as heat. So cellular respiration, so there's aerobic harvest of food and energy. So aerobic respiration consumes organic molecules and oxygen. So this is what our cells do. Cellular respiration is an aerobic process where you consume oxygen and organic molecules. Photosynthesis generates the oxygen and the organic molecules, which are used in cellular respiration, and cells use chemical energy stored in the bonds, which are broken, of organic molecules to regenerate ATP to generate work. So cellular respiration uses aerobic harvesting of chemical energy from organic fuel molecules. Although carbohydrates, fats, and proteins are consumed as fuel, it is helpful to trace cellular respiration with this sugar of glucose. So again, glucose is primarily what cellular respiration is used. The other molecules store energy, but they themselves are then broken back down into glucose to then be used for cellular respiration. So you can see in this equation, you have C6H12O6, which is glucose, plus 6 oxygen molecules, is converted through cellular respiration to 6 CO2 molecules, 6 H2O molecules, and energy, which is a combination of ATP and heat. So the transfer of electrons during chemical reactions releases energy stored in the organic molecules. And this released energy is ultimately used to synthesize ATP. So cellular respiration is broken down into 3 stages. There's glycolysis, which breaks down glucose into 2 molecules of pyruvate. That's the first step, and that occurs in the cytosol. Then there's the citric acid cycle, which completes the breakdown of glucose, converting pyruvate into carbon dioxide and water. This occurs in the mitochondria. And then you also have the electron transport chain, which accounts for the synthesis of ATP, which also occurs in the mitochondria. So again, if we look at these individually, we have glycolysis, which occurs in the cytosol, where you take glucose and convert it into 2 units of pyruvate. And in that process, you generate electron carried via NADH. So NADH is another energy source. It's a temporary energy source that the mitochondria uses to generate ATP. And then you can also directly generate ATP from this pathway. After you generate pyruvate, the pyruvate then moves into the mitochondrial matrix, which is where the citric acid cycle sits, or all the proteins and enzymes for that cycle. And when you break down pyruvate, you end up generating carbon dioxide and water, and you generate more molecules of NADH, which again are those temporarily energy sources, and FADH2, which is a similar chemical to NADH. And then you also generate a few molecules of ATP. However, most of the ATP is synthesized in the electron transport chain, where you take the NADH from both the glycolysis and the citric acid cycle and the FADH2, and use both of those molecules to convert or to generate ATP through a process known as chemiosis. And this occurs in the membrane of in the inner membrane, between the inner membrane space and the matrix. So let's look at stage 1 in more detail. So glycolysis harvests chemical energy by oxidizing glucose to pyruvate. So glycolysis, the word meaning splitting of sugar. So it breaks down glucose into 2 molecules of pyruvate. And glycolysis occurs in the cytoplasm and has 2 major phases. The first phase is energy investment phase, and the second is energy payoff phase. So this will make more sense on the next slide. So in the first phase, you have energy investment phase. So this is where you take glucose and you spend 2 units of ATP, which become 2 ADP plus 2 phosphate. So ADP is the non-energized form. ATP is the energized form. So you have to use energy to convert glucose into pyruvate in the first investment phase. And then in the second phase, the payoff phase, you can then generate 4 units of ATP. So you take 4 units of ADP and convert it to 4 units of ATP, so energy. And you take 2 units of NAD positive and convert it to 2 units of NADH. And so NADH is the energized form. NAD positive is the non-energized form. So you can see you generate 4 ATP units and 2 NADH units, as well as 2 pyruvate molecules and water. So overall, you have a net change of glucose going into 2 pyruvate plus 2 waters. You generate 4 ATP units, but you spent 2 ATP units, so you have a net gain of 2 ATP units. And then you also gain 2 NADH units plus 2 free hydrogen atoms, which would be protons. So overall, you can see that glycolysis breaks down glucose into 2 pyruvate molecules, 2 water molecules, 2 ATP units, and 2 NADH units. So in this slide, you can see how ATP is formed. So here we have the enzyme. So this purple blob is an enzyme. You have a substrate which has a phosphate group attached. So again, phosphate is that P surrounded with 4 oxygens. So it's a P atom in the center attached to 4 oxygen atoms, 2 of them through single bonds and 2 of them through double bonds. And then you have an ADP molecule. So again, the ADP molecule is the non-energized form. And so the enzyme moves the phosphate from the substrate, from this green molecule, to the ADP molecule. And when it does, that becomes an ATP molecule and becomes the energized form. And you can see that the phosphate is now missing from the substrate, and you now have a different product. So ADP stands for adenosine diphosphate, di meaning 2, phosphate meaning phosphates. Adenosine is one of the bases used in DNA synthesis. It's one of the 4 nucleotides. So adenosine diphosphate. And when you add a phosphate to ADP, you then have 3 phosphates. So it's then adenosine triphosphate. So that's where the T and the D come from. Diphosphate meaning 2 phosphate groups, T meaning 3 phosphate groups, triphosphate. And then it's ATP, which is used to generate work in the cell. All right, so the next stage is the citric acid cycle, which completes the energy-yielding oxidation of organic molecules. So this is where you break pyruvate down into carbon dioxide in the presence of oxygen. And again, this occurs in the mitochondria matrix. And before the citric acid cycle can begin, pyruvate must be converted to acetyl coenzyme A, which links the cycle to glycolysis. So again, glycolysis occurs in the cytosol, where you generate 2 pyruvate molecules. Pyruvate is then changed or converted to acetyl coenzyme A, and then acetyl coenzyme A is shuttled into the mitochondria matrix, where it can then be broken down into carbon dioxide. So this is where I will leave off for today. We will pick up on stage 2, the citric acid cycle, in class on Wednesday, September 9th. Again, Monday is a holiday, Labor Day, so we will not have class on Monday. The assignment that goes along with this lecture should be on Blackboard by the time you're viewing this recording, and is due by class time, so 2 p.m. on Wednesday, September 9th, which is when we will pick up again in class.